Great. Okay. Over the weekend, I did you guys a favor by not posting 3 sets of thought problems for translation, transcription, and genetics. I thought if I did that, you might spend the whole nice weekend working on those, so instead I've posted them this morning, along with a really cool animation of translation by Joachim Frank from the State University of New York at Albany. So those of you that learn better by looking at animations, check out the movie of Frank's animation of translation. I should also point out that all over the web you'll find marvelous animations of different things that we draw on the board here in 2 dimensions. I particularly recommend a DNA replication animation that's rather large. It's a big file that you can find at the Cold Spring Harbor Laboratory Teaching Center. So just go to Cold Spring Harbor Laboratory Teaching Center. There was a board that I promised that I would that I didn't have time to cover before, that I promised I'd try to do today. But I've decided to turn that board into some thought problems, so those you will see on one of the sheets that you'll find on the BSpace site today. So what I'll do this morning is to pick up the last part of the central dogma in preparation for our really fun discussion on Wednesday about how gene regulation works, how gene switches work, and how cells know when to turn on what gene at what time. Now, since you've already done all of your reading for today, you already know that the genetic code is a triplet code of 64 different triplets, 3 of which don't specify any amino acid, and 61 of which specify one or another of the only 20 amino acids used in biology. You already know that. That's pretty dry. That's pretty factual. So what I'll try to do today is to take you back into history to show you how it is that we actually think we've got that right. And I'll show you the experiments that led to the understanding that there was a code, how the code was read, what the code was, and then how translation works. We'll have at least 2 Nobel Prizes today. There should have been a few more, but so far only 2, and I'll describe those. So, as you already know, the code is used as a triplet dictionary, and I've drawn out a sequence of RNA here. Now, of course, the dictionary could have been much more complicated than a triplet dictionary, and it could have been read in different ways. We could have imagined a long string of letters in RNA that would cause the assembly de novo of particular protein domains or so on and so forth. And I like to think of the Hawaiian language. It has only 12 letters, a little more than 4, but you can express very complicated thoughts in Hawaiian. There's no shortage of communication. You just use big words. Now, the genetic code is a triplet code that is read 1 3-letter codon at a time, but any triplet code could be read in different reading frames. So the question is, how are we going to know where to start? And the other question is, how do we know where one word ends and another word begins? Now, what's remarkable, if you take, I've got a bit of a cold today, so we'll be struggling. If you take any 64 triplet code, you can actually design a code assigning letters to each amino acid, each of the 20 amino acids, in such that you could read that code in an overlapping fashion. That is, this triplet could spell one amino acid, this triplet could spell another amino acid, that triplet could spell another amino acid, and back to that particular reading frame. You can design such a code. Such a code always requires 64 codons, and it always requires 3 nonsense codons to have the code work. There's lots of different examples of such a code. But what we'll discuss now is how do we know that the code is read in a non-overlapping fashion? And that came from 2 lines of investigation that came together for the very first time. The first was the work of James Neal, a human geneticist in the 50s, who was able to discover, through pedigree analysis, that the common human disease among African Americans, sickle cell anemia, is inherited as a single Mendelian trait in a semi-dominant fashion. semi-dominant meaning that the heterozygotes had phenotypes that were different from the homozygous wild type or homozygous mutant. So that was a very important point. And the other point that was known at the time was that the blood, the red blood cells from people who were homozygous for sickle cell anemia, under low oxygen tension, would sickle. And that's the foundation of the pathology associated with sickle cell anemia. The cells don't go through the capillaries well. So that provided a big biochemical clue. It said that whatever the biochemical defect caused by sickle cell anemia was, it would be resident in red blood cells. Now, it turns out that red blood cells have a relatively small repertoire of different proteins. So the red blood cells from sickle cell anemia, homozygotes and heterozygotes, it was an obvious way of actually trying to get to the biochemical defect behind an inherited disease. Now, there weren't a lot of people working in this area at the time. Today, we have thousands of people engaged in biomedical work, but at the time, there wasn't very many. And the Rockefeller Foundation was concerned about that, and they decided that what they wanted to do was to get the best talent in the world working on this problem. And among the best talent in the world was Linus Pauling, probably the best chemist that ever lived. And Pauling had not really been very interested in biology. But he was very much interested in building a big research institute, which took dollars. And the Rockefeller Institute literally, the foundation literally bribed him. They said, you want an institute? You work on biology, we'll buy you an institute. Hence, Pauling got his institute. So what Pauling did was to focus on sickle cell anemia and did the following experiment. He was able to use gels to separate the contents of red blood cells in an electric field. So here is one lane in a gel, here is another lane in a gel, here's another lane in a gel, and just like the DNA gels that you've used, these protein gels separate molecules as a function of their response to the differential electric field. What Pauling was able to show by lysing the red blood cells and taking the hemoglobin from the red blood cells is that wild-type hemoglobin had this sort of a mobility. Hemoglobin from patients homozygous for the sickle cell anemia had this mobility, and heterozygotes had that pair of different mobilities. Okay? So biochemically, we would say that by this assay, sickle cell anemia is codominant because both alleles show up in the assay when looking at a heterozygote. So that sounds like a trivial step today, but at the time that was a spectacularly big step because it demonstrated the link between a mutation in a gene and a biochemically measurable defect in a polypeptide. That set the stage for a student working with Pauling, Vernon Ingram, to do the then extremely difficult task of actually sequencing the wild type and the sickle cell hemoglobin protein. It's a little bit difficult today. It was really difficult at the time. So he sequenced the protein, and he showed that there was one and only one change in the amino acid sequence in the entire protein of individuals that carried the sickle cell trait. And that was a mutation from glutamine to valine, removing a charge on the surface of hemoglobin, creating a hydrophobic patch, which caused, under the low oxygen tension, the hemoglobin proteins to aggregate in a way that caused the sickling of the cells. So that was really the very first experiment done in what we would today call biochemical genetics. Now, this single result established a very important point about the reading of the code. This result, and others like it that came shortly thereafter, showed that mutations changed one and only one protein, and therefore the code had to be read in a non-overlapping fashion. That is, AUG, if AUG is a word, then UGU could not be read as a word at this spot, but UGU would be a word someplace else. So we've gone kind of quickly over the outlines of the code. Are there any questions so far? None. Okay. Great. So that tells us in sort of an anecdotal fashion that each codon in a gene, in the messenger RNA, contributes the information for one and only one amino acid in the sequence of the mature protein. But how are the codons read? The answer to that came from really a heroic experiment from Charles Janowski, who's still active at Stanford, who established that the sequence of information in a gene and the sequence of information in a protein are collinear. This all sounds like old hat to you, but if you think about it from what was known at the time, what was known was that RNA polymerase made a linear polymer of RNA, but it was by no means obvious that the translation of that RNA message into protein would happen in a linear fashion. The experiment that established that that was true took advantage of Janowski's mutants in an enzyme called tryptophan synthase. He had many, many mutations in the gene for that enzyme, and he did what anyone would do at the time, which is to create a genetic map of those mutations. So just like last week when we figured out what the order of A, B, and D were on the mouse chromosome, Janowski did an analogous experiment involving 3-factor crosses with mutations all within the tryptophan synthetase gene and created one of the early high-resolution genetic maps. So mutant 169 mapped here at this end of the gene, 175 mapped at that end of the gene, and there was a linear order of mutations in that gene. That wasn't so surprising. It had already been established that genetic maps were linear. What Janowski then did, and to this day I marvel at how much work this took, was to purify each of the mutant proteins from each of these mutants. Not from a mutant that had all of these changes at once, but from each individual mutant. So if you think about how do you purify a mutant protein, it's pretty hard. We talked about how Kornberg purified DNA polymerase. He had an assay for the activity of that protein and was able to purify it based upon its activity involving column chromatography. But mutants that make dead proteins create special challenges. So we won't go into how he did it, but I want you to appreciate that that was really a very difficult thing to do. He then sequenced each of the mutant proteins and established that in the linear sequence of the protein, that the order of changes of each of the mutants was a perfect match to the order of mutations in the gene. So that difficult but conceptually simple experiment established the collinearity of the gene and the polypeptide encoded by that gene. He even was able to make some really interesting deductions, such as the two mutations, mutation 46 and mutation 23. These mutations mapped very, very close to each other in the gene. You can imagine that the frequency of recombination was pretty low because those two mutations both affected the same amino acid in different ways. So these, therefore, must have been two different mutations in the same codon that changed the glycine codon to the codons for glutamic acid and aspartic acid. There was another change of glycine to valine, so this gave him an idea that there was going to be a pattern by which certain codons could mutate to other codons, and we'll come back to that point in another board. Any questions so far? Questions? Okay. So the take-home message here is that translation then involves the linear reading of a message, codon by codon, guiding the assembly of a polypeptide. Okay. We'll leave that kind of analysis and take up the action on the East Coast in Bethesda in the laboratory of Marshall Nirenberg, a young guy who had just started this lab at the NIH, and he was working with a postdoc, Heinrich Mattei. Nirenberg just recently died, so until recently he was still actively engaged in science. In 1961, at a meeting of the International Union of Biochemistry in Moscow, Nirenberg described to an audience of a handful of people a really remarkable experiment. The reason there were only a handful of people in the audience was that he titled his talk, "The Dependence of Cell-Free Protein Synthesis Upon Natural and Cell-Free RNA." It's hard to imagine a more boring title for an experiment that fundamentally was the first experiment to crack the solving of the genetic code, which in its day, in the early 60s, was the fundamental challenge of all of what would become molecular biology. So what I want to do is to show you the first experiments that went to this, that ended up earning a Nobel Prize for Nirenberg, and Mattei, well, Mattei got a job in Germany, which was not a bad second-place prize. Here's how they did it. They took advantage of an assay that had been developed at Rockefeller University in Paul Zatnick's lab, which showed that if you make an extract of E. coli, so we all know what an extract is, a whole bunch of cells, the blender, the whole 9 yards, E. coli soup, and you add radioactive amino acids to that extract, what you find is that there is a combination of activities in that extract that can piece those amino acids together in such a way that it produces radioactive proteins. So that's our assay. We have an extract, and the juice in that extract can do whatever chemistry is required to take amino acids and produce proteins. Is the assay clear? The assay is fundamentally essential to understanding this experiment. Any confusion on the concept of an extract or an assay? Okay? Now here's the cool part. If you take that same extract and add immediately, instead of radioactive amino acids, you simply add time. That is, you let that extract sit in a 37-degree water bath for, half hour to an hour, and then you add your radioactive amino acids, what do you get? Zippo, nothing. Okay? So what does that mean? Well, at face value, it's a little hard to interpret that. It's a negative result. Something didn't work. You could say that all the magic juice, all the activities in that juice died while being cooked at 37 degrees C. But that, in fact, is not the explanation. What happens, and the proof of that, is the following experiment. If you take that extract, add time, incubate it at 37 degrees, wait, and then add radioactive amino acids, as well as total RNA from E. coli, then what do you get? Radioactive proteins again. So, what's happening during the wait? Well, we've talked about how RNA is chemically unstable, and we've talked about how it is important for messages to be unstable. So what's happening during this wait is all of the endogenous messenger RNA that was in that extract is decaying so that it no longer can program protein synthesis. But if we add back exogenous RNA, a mix of all of E. coli's RNAs, we make radioactive protein. Probably 1,000 different proteins. We're just measuring the production of radioactive protein. How are we doing? Any questions on that so far? Okay. That's a key concept, a really, really key concept. Because what that essentially says is that the extract is kind of like, a computer. It's simply waiting to be programmed by some operating system to do something. So the idea is that the extract could be programmed by the addition of exogenous RNA, and if you added back a simple enough RNA, then the translation of that RNA into protein might give you some clues as to how the genetic code worked. So with that in mind, Marshall Nirenberg got on a plane and came here to Berkeley, across the street, into the old Stanley Hall, where he teamed up with Wendell Stanley and Stanley's postdoc, Heinz Frankel Conrad. Now, why did they do that? Well, Wendell Stanley had earned his Nobel Prize for figuring out the structure of tobacco mosaic virus. This is the first virus that we knew the structure of. It solved the issue of whether a virus is alive or dead, that silly old question. And the important thing was that together, these guys had shown that the genome of the tobacco mosaic virus was a single strand of RNA. It was a small strand of RNA. Excuse me, there's a duck calling me on my cell phone. There. And it encoded for a very small number of proteins, primarily the coat protein of tobacco mosaic virus. Now, Frankel Conrad had dozens and dozens of mutations in the tobacco mosaic virus, and the idea that Nirenberg and Stanley had was that if they translated each of these mutants with one or another radioactive amino acid, that the pattern of incorporation of amino acids, radioactive amino acids, in different mutants could, like this pattern here, give them insight as to how the genetic code worked. So in May of 1961, Nirenberg was over across the street with Wendell Stanley trying to do these experiments. And they failed day after day after day. It took them a while before they figured out why it failed. Meanwhile, back at the ranch, Mathai, who was trained in Germany, was left with just bits and pieces of stuff to do experiments with. Control experiments which were too boring for Nirenberg to do. And because Mathai is German, he is meticulous. And because he is meticulous, everything he ever did was recorded in his notebook with precise Germanic script. And you can actually see Mathai's notebooks and figure out exactly when he did what. So on Monday, May 22nd, at 1:30 in the afternoon, he had an idea. He said, gosh, I don't have a whole lot of stuff left to do experiments with. Nirenberg got on a plane with all the good stuff, radioactive amino acids and all that. Can you imagine what the TSA would have done with him today? Anyway, what Mathai had was this completely boring polynucleotide polyribo-U. Completely biologically irrelevant. He also had 16 radioactive amino acids. Of course, he would have liked to have had 20, but they went with Nirenberg to the West Coast. But he had an idea. He said, whatever the genetic code is, U might be one of the words in the genetic code. And if so, if I program this extract with poly-U and radioactive amino acids, I ought to be able to figure out which one it is. So he did the following experiment that Monday afternoon. He added poly-U and 16 different radioactive amino acids in the same reaction, and what he got was radioactive protein. That's kind of cool. It says that UUUU, or UUU, must be the codon for one of those 16 amino acids. Okay? So what does he have to do? Well, he just has to do the same experiment 16 times, substituting one amino acid or the other to figure it out. But Nirenberg didn't leave him enough radioactive amino acids to do 16 different experiments. He had enough to do about 4. So what he did was the same experiment with 8 radioactive amino acids, and then the other 8, and then combinatorially figured it out and parsed it down from Tuesday to Friday. And by Friday afternoon, he had it narrowed down to 2 things. It had to be either tyrosine or phenylalanine. Now, At 5 o'clock in the afternoon near the NIH, you could get run over if you stand in the way between the flood of people leaving for the weekend and the labs. But Matai, this close to understanding the first word in the genetic code. Friday night in the lab, setting up the experiment, fresh extract. He's only got enough radioactivity left for this one last experiment. And here's what he did. He set up the experiment, he translated, he took the material from those two different translation reactions, put it in a scintillation counter, which counts the amount of radioactivity by the decay of the radioactive particles in counts per minute. And being German, he does all kinds of controls. And what he found is that if he programmed the extract with poly A and radioactive tyrosine, he observed 60 counts per minute. Whereas if he programmed it with radioactive phenylalanine, he got 50 counts per minute. Not many counts, trivial little difference, that must be background. Poly C, 45 and 38. And at 3 o'clock in the morning, that Saturday, the poly U data showed him that with tyrosine it was 70 counts per minute, and with phenylalanine, 38,800. Right? Convincing proof that UUU is the genetic word for coding phenylalanine. Isn't that amazing? All by himself, 3 o'clock in the morning. You got to tell somebody. He probably tracked down a janitor to tell him. You can imagine how impressed they were. So we know that poly U is phenylalanine. Yes, sir? Oh, the question is, why does it have to be a triplet code? So to finesse that, I can tell you that your book says it's a triplet code. I can give you a logic that if you had a two-letter code, there wouldn't be enough code to establish the code for 20 different amino acids. A three-letter code is enough to do that. But if you're wondering why it wasn't a 4, 5, or 6-letter code, there was a wonderful experiment that we won't have time to go into by Sidney Brenner and Francis Crick called, if you want to look it up, it's a very famous experiment called the Aunts and Uncles experiment that establishes experimentally the triplet nature of the code. We'll just use arithmetic considerations to say that that was the most reasonable code. And you're right, these data would be compatible with a 3, 4, 5, 6U word to the code. Okay? Good question, thank you. Other questions? So shortly thereafter, Nuremberg discovered, well, he came back after having failed miserably to translate TMV, and the reason is that the genome of TMV is a single-stranded RNA, but it's not the strand that is translated to make the TMV proteins. It's the complement of that strand. The experiment was doomed to failure. In about 8 minutes, you should be very suspicious of why poly U was translatable in this extract, and I'll come back to explain why that was at that time. So shortly thereafter, the rest of the codons were assigned by another person in Nuremberg's lab, Phil Leader, by a different method called a filter binding method, which then experimentally established that it was a triplet code because they used 3-letter long pieces of RNA in that assay. I won't give you the details of that now. You'll get that in MCB 110. Instead, what we'll do is to move on to how it is that proteins are actually synthesized from the information in the messenger RNA. So the issue will be peptide bond formation first from a chemical perspective and then from a molecular biological experiment perspective. So in green is one amino acid, and in blue is another amino acid joined here by the famous peptide bond that Jennifer told you about earlier in the semester. Notice that there are 1, 2, 3 bonds in each amino acid about which molecules can rotate, which allow us to have the fantastic complex folding of polypeptide chains into the three-dimensional structure of the proteins. There is a carbonyl group at one end, an amino group at the other. So all proteins linked by linear arrays of peptide bonds have one free amino and one free carboxyl. Now, our challenge in making a peptide bond in a living organism is that it is energetically unfavorable. So you've seen this solution before. If you have an energetically unfavorable reaction, the way to make it go faster is to create an intermediate that is reactive. So that's the guiding principle by how we provide the energy for peptide bond formation. Where the catalysis comes from, we'll get to shortly. So generically, the way we do that is to connect an amino acid to an intermediate with a high energy state that is produced by the consumption of ATP. There's a couple intermediates in this process that we'll get to at MCB 100, but for now just know that we need to create a chemically unstable intermediate using energy from ATP to connect an amino acid to this intermediate in protein synthesis called a transfer RNA. So this green structure, drawn like a clover leaf, represents an RNA molecule that is somewhere between 75 and 100 nucleotides long, that spontaneously folds into this shape due to intramolecular hydrogen bonds between the bases here and the bases there, the bases here, the bases there, the bases here, and the bases there. The first structure of a tRNA was worked out by none other than Berkeley's Sung-Ho Kim through X-ray crystallography. Like all RNAs, it has a 5' and a 3' end. There are about 45 different species of RNAs in a cell, in an E. coli cell. It varies slightly from organism to organism, which then read the 61 different sense codons in the genetic dictionary. And each and every one of these tRNAs has the very same sequence at its 3' end, CCA. And that's an important recognition determinant, along with the structure of individual tRNAs, for a class of enzymes called amino-acyl tRNA synthetases. There are 20 of these in a cell, 20 different ones. Each of these is dedicated to linking an amino acid, a specific amino acid, to the 3' end of all of the different tRNAs that are in charge of introducing that amino acid during translation. Okay? So let's pause and think about the numbers here. 20 different amino-acyl tRNA synthetases, 45 different tRNAs. So what must be true about the specificity of an amino-acyl tRNA synthetase? What must be true? What do you think? We've got to put 20 amino acids specifically onto each of 45 different tRNAs by 20 amino-acyl tRNA synthetases. What must be true? Some of these have got to be utility infielders at least, right? Some amino-acyl tRNA synthetases must be able to put the same amino acid onto more than one tRNA, or the numbers don't work, right? What else must be true? There are 64 possible triplet codons. You know that 3 of them are nonsense, therefore 61 are sense, and they have to be translated by 45 tRNAs. So what must be true? There must be a few tRNAs that can translate more than one codon, right? Is the logic evident now? Okay. I'll show you why that's true in just a minute. So the big deal here is that we spend energy through an enzyme, a family of enzymes, really smart enzymes that figure out how to put one amino acid onto several different tRNAs here at the 3' end in a linkage that is primed to make a peptide bond. And here is the link of that amino acid to the 3' hydroxyl of the A at the 3' end of the tRNA. You will all recognize this as being ribo-A. That's the A up there at the 3' end of that tRNA. So that is the critical intermediate in translation. Now, translation is carried out by a gigantic ribonucleoprotein called the ribosome. It is the most complicated structure in biology ever to be solved by X-ray crystallography. Jamie Cate, a professor here in UC Berkeley, did this while he was working in Tom Steitz's lab at Yale. Steitz got the Nobel Prize. Noller, our colleague at UC Santa Cruz, Harry Noller, figured out that in the ribosome, take-home message down here in pink, that RNA does the catalysis. This is another example of the RNA actually being the enzymatic activity which makes a peptide bond. So those are the big messages about ribosomes. Let's decompact what ribosomes are all about. Prokaryotic ribosomes and eukaryotic ribosomes are similar in flavor. The prokaryotic ones are slightly smaller, slightly simpler, and much better understood. So I'm going to discuss only prokaryotic ribosomes and only prokaryotic translation. The themes are the same in eukaryotes, but there are many more parts with many more names to get confused by. So we'll concentrate only on prokaryotic ones. Prokaryotic ribosomes come in a large and a small subunit. The large subunit has a size called 50S, referring to Svedberg units, not particularly important. What is important is that the large subunit has two different RNA molecules, a 23S and a 5S, and 39 different proteins. The small subunit has its own RNA, 16S RNA, and 24 different proteins. So for decades, the enormous complexity of the protein complement of ribosomes had all of us thinking that it was surely the proteins in the ribosome that were doing the chemistry, and the RNA was just there for structure. That view is completely wrong, thanks to the discoveries of Harry Noller. We now know it is the RNA that is the catalyst. Just as an aside, the 16S RNA is the same 16S RNA whose sequence was the foundation of the phylogenetic analysis that you guys did in the BIO101a lab with Vibrio. We use 16S ribosomal RNAs to build phylogenetic trees, primarily because we know that in every single species, this RNA does the same job, whereas proteins, for example, can have day jobs and night jobs and vary in their function in different species. 16S ribosomal RNA does not, and that's why it's the foundation for so much phylogenetics. Okay, let's talk about how proteins then actually get made. The first step, called cleverly initiation, takes place when a messenger RNA, the small subunit of the ribosome, and a tRNA specifically for methionine come together in such a way that the tRNA's loop here, called the anticodon loop, pairs with the AUG codon for in the messenger RNA. So every single protein, with very few rare exceptions, begins its life with a methionine as its very first amino acid. There is something called the ribosome binding site that guides the 30S subunit to sit down in such a way that the AUG codon for methionine and the CAU anticodon in the tRNA come into register, so the hydrogen bonds between the bases position the tRNA appropriately. So all proteins start with AUG, which is decoded by a methionine tRNA. That step is followed through the agency of initiation factors, IF1, IF2, IF3, and a bunch of GTP to create the following assembled ribosome. Small subunit, big subunit. Here's the methionine tRNA, base paired with the AUG. And note that the pairing of the tRNA with the message follows the same anti-parallel rules that we've seen for every nucleic acid hybridization so far. Okay? So from here to here is the initiation step. Something that you'll see throughout translation is, for reasons that are still obscure to me, I don't know why, the vast majority of the energy in the translation process is provided by GTP instead of ATP. The reason for that is probably buried in the obscurity of the origins of life back in the RNA world. I'd like to know, but I don't know why GTP is the major form other than in the charging of the tRNA. So what is evident now from the crystal structure of the ribosome is that there are sites or pockets in which different things fit. So there is a site called the P site for the peptidyl site, where the first charged tRNA sits. There is a E site for ejector site or empty site. And there is another site called the A site. The formation of the intact ribosome is done in such a way that the very next codon is at the A site. So the reading of the frame in the message is provided by the three-dimensional structure of the ribosome. There's no spacer in between. Right here is GUG, which is then recognized by the tRNA, whose anticodon is the complement of that. In this particular case, it would be alanine. Okay? So the alanyl tRNA will come into the acceptor site. Any questions so far? Each of these steps is individually very simple, very straightforward. It's the aggregate that gets to be a little bit confusing. No questions? Okay. Let's pause for a second to emphasize 5' to 3'. So in the anticodon loop, the 5'C base pairs with the 3'G in the codon. The middle A pairs with the U, and the U pairs with the A. It turns out that because of the structure of the ribosome and the tRNA, the Watson-Crick base pairing rules for the 3' position of the codon and the 5' position of the anticodon are a little loosey-goosey. They're called the wobble rules that is the chemical foundation for how it is that 45 tRNAs can decode 61 codons. We won't worry about the hydrogen bonding that takes place in the so-called wobble pairing, but when you work on the concept problems and study deeply the structure of the genetic code, you will notice that in most cases, the third position in a codon can change to anything without changing the identity of the amino acid that that codon spells. That's a general rule. The third base of a codon is not under very much selection. Okay. So the next step in protein synthesis is the formation of the first peptide bond, so the alanine tRNA base pairs here. And now what we have is the amino group on the alanine attacking the carboxyl group of the methionine, breaking the linkage of the methionine to its tRNA, and transferring that methionine covalently to the alanine, which is covalently attached to the tRNA in the A site. Okay? This defines the direction of protein synthesis as being in a amino to carboxyl direction. Okay. So what happens then? Well, we've got a plugged A site. We've got something useless at the P site. We can't continue unless we move stuff around. So we'll burn a bunch of GTP and engage the action of 3 different elongation factors, EFG, EFTU, EFTS. Don't worry about the details. Know that there are elongation factors. They're enormously abundant cells in the cell. And as a result of that burning of the GTP, what we end up with is the transfer of the methionine tRNA, now spent, into the ejector seat. Whoosh, it's gone. We have the growing polypeptide chain here, and the next codon is open and available for a phenylalanine tRNA to direct it to that position. Okay? So that's a step in protein translation. And from here to there, it's about 1/10 of a second. Okay? It's really quite fast. Okay? Pretty straightforward, huh? So how does it all end? Well, it all ends with stop codons. Okay? Stop codons are those 3 codons in the genetic code that do not specify an amino acid. They're sometimes called nonsense codons. And mutations in genes that create stop codons are referred to as nonsense mutations. There are 3 stop codons, UAA, UAG, UGA. You may see these referred to as the ochre, amber, and opal codons. If you do, just know that those are the names of particular stop codons. The names are not that important. But let's look at what happens when one of the stop codons appears in the A site. At that point, there is no tRNA that can decode it. Instead, that 3 letters is read and recognized by a protein called releasing factor. So what releasing factor does is to actually change the peptide bond formation chemistry. Now, the attack on the carboxyl group of the growing polypeptide chain is done not by the amino group of the next amino acid, but by water. So essentially, we transfer the growing polypeptide chain by the catalysis of the ribosome onto water. And at that point, boom, our polypeptide is released and folds into its 3-dimensional structure. We then have an empty ribosome. Guess what? There's yet another factor that, guess what, burns more GDP that disassembles the whole thing so they can be recycled. Okay? Now, one of the things that's really important about understanding prokaryotic translation is that it is the target of many, many, many antibiotics. Okay? And in fact, the 3-dimensional structure of the ribosome is so exciting to the world because it may now be possible to use rational drug design to find out yet more ways of exploiting the vulnerability that comes because all bacteria have a relatively different ribosome from eukaryotes. So among the drugs which many of you have experienced already, tetracycline, right? Tetracycline works to kill bacteria by blocking the A site. So tetracycline would occupy that A site and no tRNA could have access to it. The bacteria will die. Chloramphenicol blocks peptide bond formation, so it freezes the catalysis of the ribosomal RNA. and one of my favorites in terms of its name, kasugamycin, specifically blocks the association of the initiating methionine tRNA with the ribosome. So that's the outlines of protein synthesis. But what I want you to think about now is the limitations of life as we know it. Biology uses 20 amino acids. If you think about the R groups on those 20 amino acids, you will discover that they have a very narrow selection of chemical reactivities. Nothing compared to what your organic chemistry friends use routinely. So what if we could expand the library of life? Synthetic biology is a field, a brand new field, that is designing efforts in exactly that direction to create life which has an expanded repertoire of amino acids. If you were an organic chemist, you would want to build amino acids with keto groups or aldehyde groups to be able to do novel chemistries. Think about what it would take to add an amino acid to an E. coli cell with an R group, which is a keto group, and get it incorporated into protein. That's already been done by one of our colleagues here in chemistry, who was here in chemistry, Pete Schultz. We can now make non-natural proteins in E. coli cells. Think about what he had to do to make that experiment work. And on Wednesday, we'll talk about prokaryotic gene regulation.